Hello, my name is Jihoon Choi from We Learn to Share. Today, I'd like to discuss how neurons work. So, this is a diagram of the brain that I drew. Um, I'm a humble person, but I would like to admit that this is a pretty good drawing. Anyways, inside our brain, there are neurons, and there are neurons in our body too, and there are just in the brain 86 billion of them. I'm gonna spin it to emphasize that. One neuron, what it does is that it processes information and it delivers that information. But what happens when there are multiple neurons? When they connect to other neurons, the real magic happens. It gives rise to thoughts about thoughts, and to thoughts about the thought that was thought. How cool is that? So I think it's important to realize the mechanism behind how a single neuron can operate to better understand how it acts as a collective group. So this is the basic structure of the neuron. There's the dendrite, which receives signals from other neurons. There's the cell body, which doesn't really do anything except supplying neurons with nutrients and having DNA and all that. The axons send results to other neurons, and these tips of the axons will connect to another dendrite of another neuron. And as you can see, there are multiple axons. What this means is that one neuron, the results of it, the result can be sent to many other neurons. This is a specialty of our human brain. There are trillions and trillions of connections, and I think that's really cool. So when did the study of neurons start? It started by this guy Luigi Galvani, and basically he put electricity on a dead frog's leg, and it started moving. So he named this term animal electricity. But it wasn't until these two people, who got the Nobel Prize, that we really know how neurons work. We knew that it operated on some electricity, but it wasn't until these two actually came up with a science theory of how it deals up with electricity, our neurons. And so we're going to be studying the mechanisms underlying the electricity in our neurons. So let us go deeper into the dendrite. In our dendrites, I think it's important to clarify the intracellular parts and the extracellular parts. So the extracellular parts are the white parts here, all the white part, except this wide circle here. I'm supposed to color it, but I was too lazy. Anyways, this outside part is called the extracellular part. The intracellular, on the other hand, is the name given to the green part, okay? And also this part here for the axon, because axons also use the same mechanism as the dendrite. So the intracellular is the green part here, extracellular outside. Okay, so now that we got the distinction, we can go straight to the mechanism. So basically in the dendrite and the axon, there are two main ions. There's the potassium and there's the sodium. This is the intracellular part, extracellular part. And there's the cell membrane. What the cell membrane can do is it blocks sodium, the Na, from going into the K. So this is a theory in science, or a very well-known a fact that things want to go from many to less. So the sodium here, as you can see, there are more of them. And in our bodies, there are a lot more sodiums outside than potassium in the inside. So they want to go in, right? Because many want to go less. So when they're trying to go in, like when they're bouncing off but they're when they try to go in, they're bouncing off the cell membrane because the cell membrane is like, nah, bro. They're like, it's too crowded, let us in. Nah, bro. So the cell membrane kind of creates this electrical imbalance, I should say, between the intracellular part and the extracellular part. So what it does is that it creates more positivity outside and more negativity inside. So you might see, oh, but potassium is also plus. So shouldn't it be a plus sign? Well, I'm talking in terms of relative amounts here. So relatively, there's less potassium than sodium. And this is an important concept to remember because this is the concept for voltage. So voltage is not an absolute value, it's more like a relative value. So it's saying the inside is more negative than the outside. And in this case, millivolts is in terms of the inside compared to the outside. But not all hope is lost for sodiums. There are these things called ion channels that open and close. So currently here, there's none. So sodiums cannot go in. But in mechanisms which I will discuss, um, some of them are allowed to go in and they can be happy again. And these are called ion channels that allow sodium to go in or potassium to go out or in. There are other types of ion channels, but I just drew it for sodium. Okay, now let's get to the real facts. So there are neurotransmitters. I'm sure you've heard of them. One of them is glutamate, and they are released from the axon of the previous neuron. So as I talked earlier, these axons are connected to other dendrites. So the axon here sends neurotransmitter to the dendrite of the other neuron or to the next neuron. And one of them is called glutamate. In our body, 80% of our neurotransmitters are excitatory. And such neurotransmitters include glutamate. So what it does is that it binds to these receptors in the dendrite, and they are actually ion channels. What it does is it opens a gateway for sodium ions to go into the cell. As I said earlier, the sodiums really want to go in. So when the glutamate fits into the receptor, they're like, oh yay, it's open, and they're going in. You don't have to worry about them going out because this is biologically made to only allow them to go in. This is a graph that I'll be using for my explanations. Millivolts, as I explained earlier, is the relative amount, right? And then there is more negativity inside the neuron. So it creates a negative millivolts or a negative charge inside, and it stays at 70. This is called the membrane equilibrium. But what happens when the sodium are allowed to go in? There is more positivity inside. So as you can see, the millivolts start rising and rising because the sodium channels are starting to open. And they're like, yeah, baby. They're really happy about that. Bad news for potassiums. They want to stay alone, but unfortunately, the sodium's channels open as soon as it reaches negative 55 millivolts. These are called voltage gated sodium channels. They look different. And basically, it is there's a lot of these voltage gated channels for sodium relative to these ones. So when enough of these are stimulated, when there's many glutamate, the Na comes in, and when it reaches negative 55 millivolts, a lot of them opens because of this voltage gated channels. They open at a specific voltage, in this case negative 55 millivolts. So Na ions come in, and K plus ions are very, very angry about that. They want to stay alone. But when it reaches 40, it changes. When it reaches 40 millivolts, the sodium ions close, because as I said, it's a voltage gated channel. Instead, potassium's ion channels open and they go out. So for them, it's revenge time for coming into their territory, and the Na ions can't go in anymore. This causes more negativity again because there's an influx of the positive outside. So basically, if we imagine that there are ion channels along the dendrite like this, of course it's not to scale, the electrical thing, like this signal for all of this, I don't know, parts, these parts here and here, it's going to create a cascade of these electrical stimulus, and this is called the action potential. And the action potentials continue down to the axon, and in the axon it's going to stimulate the neurotransmitters to be released to the next neuron, and so on. So this is some extra information. As I said, 80% are excitatory. They try to stimulate this uh this this positivity of the intracellular membrane. But there are other neurotransmitters such as GABA that try to make this go down, like as you can see here. What they do is they open chloride ion channels, which are called AMPA and NMDA, and basically there are more negatives inside, so it's going to kind of resist a change in glutamate levels, and it's going to inhibit the neuron. Another fun fact, or another fact, I guess, is that there's a constant sodium potassium pump. It sends 3 sodiums out and 2 potassium in. So you might say, oh, why this imbalance? Well, as I said earlier, neurons act on the millivolts, the voltage, the wanting of sodium to come into the cell. So it needs to have many sodium ions outside compared to the inside, so that it's less positive on the inside compared to the outside. Since we learned about the mechanisms of neurons, I think it's also important to wonder about this question. Are we just a computer if we're biological? You can think about this yourself, but because I don't have a clear answer. A computer can be defined in many ways and so on, but if you're defining computer as just a chip, I think that is not true. Our brain, our neurons, have the ability to mold itself. It's able to constantly change itself. Even though you may think it's biological, these changes cannot happen in computers or whatever. And that is a fascinating thing about life. It constantly changes. I hope that this lecture was able to appreciate what nature has to offer. If you want, you can write down in the comments what you think about this question. Anyways, thanks for watching, and I'll see you next time. Thank you.